We're live.
All right. What's up, everyone? Today is opposite day, as you can tell, because I am your host. Let's get this going. This is going to be a fun one. Today's debate is on pigs can fly. Actually, no, wait, that changed, didn't it? It is there evidence for human evolution? So we have a fun debate. And tomorrow is actually another debate. Is oneness biblical at 6:30 p.m. Pacific Standard Time? But for right now, on to our debaters. First, we have Standing for Truth, the man, the myth, the legend, who needs no introduction. And we also have the rising star, the challenger himself, Luca. And before we get into going over the scenario, what's going to play out, let's ask some of the debaters here to introduce themselves just briefly and anything that they want to say. Introduce yourselves and your channel, any websites that you might have. And we will begin with Standing for Truth.
Well, thank you, Matt. Great introduction. You're going to be taking my job soon. It was so good. So I appreciate it. And yeah, I'm Donny, or many may know me as Standing for Truth or SFT. So this will be my, I believe, debate number when it comes to formal debates, number 96. And I love these topics. Luca, I appreciate him doing this debate. He's had several now, and every debate that he does, he comes prepared. He does some fantastic prep work. It brings slides, technical papers. So I'm really looking forward to this. I think this is going to be a great debate. Like I said, this will be my 96th debate, and I've had debates with some well-known evolutionists and PhDs like Dr. Stefan Frello, Dr. Dan Stern Cardinal. I've debated Gutsy Gibbon 3 times, I believe, and Tom Jump a couple times, a Conspiracy Cats a couple times. So yeah, I've been doing this for about 3 years, and I've got a series of books as well, with a few of them behind me. And as a ministry, me and Matt are working on a human evolution handbook, hoping to get that out in the next few months. So this is the perfect debate, since that's the book we're working on right now. So Matt, I appreciate it, brother.
Awesome. And Luca.
Yeah, I'm Luca. I'm an atheist, and I've been there for quite some time. I think I was around since the beginning of this channel, when it was much smaller. So you know me from quite some time, both Donny and you, I think. And yes, I did some debates. The first one was with Kent, was quite a blast. I have to say that. And I come in this debate prepare as it was possible. It's not my field. I'm a chemist. I work as for now as a teacher. So it will be tough for me, but I did my best. My knowledge on the subject of today was derived by direct experience during my high school years. I did a lot, and we'll talk about it later. So I hope to have fun with this debate. It's always good to be here, and let's go.
All right. Okay, everyone. We are going to, I wanted to remind everybody to be sure to tag me. If not, it doesn't really matter. I'll be watching for questions most of the time. Just put a lot of question marks. I can't miss that. That's great because they come up colorful. That really helps me. And for the debate, what's going to be happening is we're going to have a 17-minute openings. And if there's extra time, we might go into it later. We might leave it for the open discussions or even Q&A, but it's very early in the morning for Luca, so we probably might close it up a little bit early just for that. But after that, then we have 10-minute rebuttals to those openings each, and then we have a 30-minute open discussion with 5-minute closing from conclusions from each person. And then we jump into a 25-minute Q&A. So again, be sure to leave any kind of questions for the debaters to myself, or standing for truth, or just make sure it has a lot of question marks so I see it. And don't forget, there is going to be an after show on Logical, Probable, Possible, which is John's channel. I'm sure if he's not here, he will be here soon. Actually, I think I already see him. There we go. And so, yeah, he'll be in the chat leaving links to where that after show will be. And now we will jump right into it. Who is going first? Luca. Okay, awesome. I will be timing it. Just to let you know, in case you get near the 17 minutes, which is quite a long opening. So that would be impressive to go 17 minutes, but I will do it nonetheless. And
I will try.
Whenever you are ready, if you want to share screen, I can't help you there, but
I got you, Luca.
Okay. Awesome. And whenever you are ready.
Yes.
As Luca's getting that ready, Luca, once I see it pop up, I'll put it up on screen. Berean Perspective Apologetics says, a cup of coffee on me. Lord bless. Lord bless you as well. You know how much, at least I love coffee, so I appreciate that. And Logical, Plausible, Possible, as Matt, you like to say. He's got his dumpster fire guaranteed to be dumpster fire, I'm sure, after show. So he'll be advertising for that.
Okay, you can see my
I see it now.
Okay.
Yeah, you're good.
Luca, I saw a comment by Cheryl in the comment section that says you're hard to hear. Did you want to make a couple comments and see what the audience says? I want them to be able to hear you. I hear you.
Can you all hear me? We are ready.
Yeah, I can hear you fine. You sound
yes.
You sound good to me. You coming in loud.
Fine here too. Okay, I was just making sure.
Okay, if you are ready, I can start my presentation.
Okay, whenever you're ready.
Okay, so there is evidence for human evolution. I think so, of course. Let's talk about human and apes first. The idea that we are related or in the same category of the apes, it's not from Darwin, and it's quite important because the first one was Carl Linnaeus. It's quite famous, and of course is much older. It's a much older work than Darwin. It's important because these ideas was not. It does not originate with Darwin, but it was precedent that we and the apes are the same thing, basically. And he was a creationist. It's quite important, this thing. So, humans are apes, and I do think so. But what I mean with that, it's not an insult. I'm not trying to be rude or anything, but I'm just pointing out that we are in the same category of apes. You all can see the skeletons of some apes and the human, and we do have the same characteristic. We are in the same superfamily and even in the same family with other apes like Pongo, gorilla, and so on and so forth. But I've been on this channel for quite some time, so I know that it's not nearly enough to convince you. And I can understand that, because you can say to me that we were just we are in this way because we were done by someone. We were designed by someone in this way. And it's possible. I don't think so, but I can understand that point. So there is evidence for human evolution. Yes, there are quite some line of evidence for it. And you cannot find those evidence just in one field of science, but in several. But there is a problem. As I said, it's not so easy to convince someone that there is the case. And I can understand that. So we can point out similarities, we can talk about vestigial structure or fossils. That's another big one. But no one will be convinced by these things. I do not have any expertise in those fields, and I cannot talk about these things with any confidence. But as I said, anything in these fields that I show to you will be not enough to convince you, I think. So let's talk about retrovirus. Endogenous retrovirus. It's a topic done on this channel in quite some videos at that. So I'm sure that you do know this topic, and I'm sure that you do know it quite well. So why I want to talk about these things? Because I had some experience during my high school years. Those are direct experience, and it was very hard to find those evidence that I saw during my high school years, because I found out that there are very, very specific things. I had to go in some papers to find those things. So let's talk about restriction enzymes. Why are they important? Why and how they work? Those enzymes are enzymes that can cut the DNA. To cut the DNA, you need to do it in a certain way. Why? Because if you do so with, and I think that you all can see where my pointer is, if you do a clean cut, the DNA will be useless. You will be no longer able to reattach the DNA. To do so, you need to do an uneven cut, to cut this way, basically. Because if you do so, you can reattach the DNA with those parts. They are called sticky ends because they can bind in these points, and then you can reattach the DNA. So they are used by viruses and by bacteria to cut the DNA and insert their DNA to use the cell to their advantage. And in this case, we are talking about virus. The results of this process are 2 regions of DNA for each virus, endogenous retrovirus, called RRTs. And that's quite important, as we will see now. This is my site. Okay, endogenous retrovirus, what they are. Here you can see a scheme about this kind of virus. And here you have a quite quick explanation about this. Those are retrovirus and exogenous retrovirus inserted that they had inserted themselves inside the DNA of the host, of their host. Sorry. As you can see it, there are two regions at the end, at the beginning of the sequence, called LRTs. That there are no coding. They are basically useless. To use figure of speech, they are like scars in the DNA. Inside those two regions, you can find the regions that can produce some proteins for the virus. Here you can see the kind of proteins, structural proteins for the virus. Of course, the ones inside our DNA are deactivated for the most part, or they are modified in a way or in another to be useful for us. Some are even very, very useful, but the vast majority are just there. So, how we find those kind of things in our genome? Here you can see again the structure and the result of a specific test that you can run. The test is called BLAST, and it's a specific test to find some kind of repeat in the DNA sequence. You put a sequence inside the machinery and you will find those sequences. What kind of sequence? The LRTs. You can find them, and when you find them, you can run more analysis and find out where the retrovirus are in our genome. When you found those sequences, you can compare them with whole other animals. We can compare our genome and our RNAs inside our genome with other species like apes. It's a quite important process because those kind of sequence are remnants, are basically scars that we got from an infection. Our ancestors got an infection when they were quite different from us. They were something else. It's something that happens in the past. And we can compare those sequences and find out where we diverge from other species. If we have the same set of scars, we know that we have a common history with that species, and so on and so forth, until we can build an actual tree, the famous lines of paper on paper. Sorry. So it's a very specific process. I actually tinkered with it during my experience, even though I was just in high school. So what are the results? Here you can see my sources. Okay, what are the results of this process? And it's a very good process, if you ask me. You can run those tests right here, right now. And here you can see us related to various apes. And here you can see, all of you can see different trees, because we can analyze different genes, but more or less are the same. So with those analyses, we can link our species to others, like apes, orangutans, and other humans, gorillas, chimpanzees, bonobo, and so on and so forth. And we are not talking about things in the past. Of course, those infections were in the past. No matter what's the model, it's still something happened in the past. But we can perform those experiences right now. And those are the results. So that's my whole point. It's very important to me to explain to you all these kind of things, because I did learn about those things quite some time ago, and it was quite experience for me. So that's the end of my presentation. I just want to inform you that I did study quite a lot on this subject. Those are my old books from my high school years. They were very useful, if you ask me. For high school books, they were quite informative. I went to a quite good school, if you ask me. And I hope that you all enjoyed the presentation. Here you can find my mail. It's quite important if you have some questions. I do love to engage in those kind of discussion. I bet that a lot of you know that already. And thanks for watching. And I want to give my thanks to a special person, Chantal, a friend of mine that helped me a lot with this presentation. Without her, I don't know how I would done it. She helped me a lot. So I'm done.
Oh, Matt, I think you're on. You're good now, Matt. You're good now.
All right, thanks. I didn't even know. Yeah, good job, man. You went over 16 minutes, so perfect.
Okay.
You almost used up your whole time. Great. Awesome. So now, Donny, whenever you are ready, unless you had anything to tell the audience or ask, go ahead.
No, I'm good. Is my audio video coming in good?
Sounds good to me.
All right. All right, here we go. Let me just share screen here real quick. Luca, thanks for that opening statement. I appreciate it. As I said earlier, I mean, you come prepped, you come with some good, interesting arguments, and I am very excited to engage them. So let's see. I imagine these slides are showing up on screen. So that being said, let's get right into it. I'll start the timer now. 17 minutes. Debate number 96, human evolution. Is there scientific evidence for human evolution? Donny versus Luca. I'm excited for this. As I said earlier in the intro, I've written numerous books with a future one specifically on this topic coming out. Now, it's important to understand that fossils are limited in terms of what they can tell us. And I do appreciate that Luca spent some time on the genetics data, and we will deal with that in the rebuttal. And this is why the fossil record is low quality and low confidence science, because of the limited data that we can really get from it. It requires huge amounts of assumptions, inferences, and imagination. Genetics is the best way to answer this question of ancestry. Is a fossil inherited sperm and egg? No. Genes and traits are inherited sperm and egg. You can only do so much with fossils. Ultimately, if we want to answer this question of ancestry, we must go down to the molecular level. Most of the hominids do not have DNA that can help us determine what they were like and to see how close or how far they may have been from us today. Their DNA could tell us a lot about the world that they lived in and how they lived. This is why Neanderthal DNA has helped us greatly in understanding who they were and whether they provide evidence for human evolution or separate ancestry. Firstly, genetic data has revealed that we, as humans today, have mated with the so-called ancient humans. These would be Neanderthals and Denisovans. It is understood that roughly 4% of the DNA of European modern humans is shared with Neanderthals. Again, this means modern day humans have interbred with Neanderthals and the Denisovans. Was this predicted by the evolutionary model? Of course not. Have a look at what was once assumed to be Neanderthal by the evolutionary community, and this was based on fossil data without DNA. Earlier reconstructions portray Neanderthals as a brutish subhuman species. You can see this on the left. Newer museum displays are replacing the outdated representations. In keeping with the latest DNA analyses, Neanderthal replacement displays appear more modern in appearance, and you can see that on the right side. Neanderthals were massive. As you can see here in the image, they were robust and built for the cold post-flood ice age related conditions they lived in. Their musculature and bone structure were giant. I was watching a lecture where the speaker explained that nearly all Neanderthal fossils that you find have broken bones somewhere in their body. These are bones that broke and then healed. We see the evidence of this in both Neanderthal men and women. That means they were hunting together, they were working together, and we know they lived in cruel, cold environments where they wrestled and killed animals for food. They lived in small populations, and as a result, they were inbred, highly inbred. Their rungs of homozygosity are massive. They had better eyesight than us, bigger brains than us, and they were stronger than us. And experts in the field would now admit that they were the same species as us. We literally have their genes. Okay, they were not a different species, not a sister species like some evolutionists still want to maintain. They were the same species. A ton of interesting information on Neanderthals. For example, right here, inbred Neanderthals left humans a genetic burden. This is all exactly what we'd expect from the biblical creation model of ancestry, where you have these subpopulations migrating after Babel, or maybe even some before, and they become isolated, inbred, and mutations accumulate. Mutations manifest, the ones especially that are in a recessive form. Notice this: inbred Neanderthals left humans a genetic burden. The genetic cost of Neanderthal introgression. Famous sites such as the Pit of Bones in Spain show that it is possible for a single population, we find this in one single Pit of Bones, guys, to display features characteristic of all major hominin species in the genus Homo. You got Erectus, Heidelbergensis, Neanderthals, and Homo sapiens. This proves that all these people coexisted and were part of a diverse interbreeding population. Notice this, guys. This stuff is fascinating. Neanderthal behavioral practices and abilities. Manufacture of pitch, controlled use of fire, sewing, tailored clothing and footwear, living floors, cosmetics, cared for the injured, weak, elderly, ceremonial burial, abstract thinking. I mean, this was a very sophisticated and intelligent people group. Evolutionists would admit that even according to their own model, humans have shared the planet with multiple different species of human, as we've seen here in the Pit of Bones. We've got, what they say are different species of human living at the same time, found together in the same bone pits. Okay, and they would say that these would include Neanderthals, Heidelbergensis, Erectus, Naledi, Denisovans, Luzonensis, and Floresiensis, who people might understand as the Hobbit. Paleoanthropologists would also admit that what they would refer to as a different species have coexisted with the Australopithecines. What we see in the fossil record, and I say record very loosely, is extensive overlap. We see coexistence and overlap. This is exactly what the biblical creation model of ancestry would predict. Evolutionists, unfortunately, have had to reject this linear idea of human evolution because of what we see. Notice this from Donald Johanson himself. The collection suggests that Homo and Australopithecus coexisted as early as 3 million years ago. The coexistence of Homo and Australopithecus in early South African sites it's not even a rare occurrence. It's not unusual. Notice this. This is a technical article by Ella Bin and Yoel Rak. The lumbar spine of Australopithecus sediba indicates 2 hominid taxa. Again, the coexistence of these different genera is not unusual. However, a careful assessment of the fossil remains attributed to Australopithecus sediba lead us to conclude that is in other South African caves. Notice other South African caves. Okay. This isn't some minority thing. 2 species are present. One represents Australopithecus and the other Homo. You don't really hear about this in the textbooks, do you? In groundbreaking find, 3 kinds of early humans unearthed living together in South Africa. Notice this. 2 million years ago, according to their own time frame, 3 different early humans, Australopithecus, Paranthropus, and the earliest known Homo erectus, which you're going to see in a bit was completely human in every way, appear to have lived at the same time in the same place. All right, so this is what we see. And you will see by the end of this presentation and debate that there were not different species of human. You will see that we are not related to the great apes, and we have not descended from Australopithecine or ape-like ancestors. Notice this phylogeny. That's right. We can confidently determine what is and what isn't related to humans. And we can do this with the genetic related data. For example, the Y chromosome, the mitochondrial DNA, the fact that we have low genetic diversity. We could look to DNA function. We can look to endogenous retroviruses, mutation rates. We can look to linkage blocks, tons of genetic related data that separate us from the great apes. Notice here the separate ancestry model. Listen, the fossils that we have for human evolution are either fully human, degenerate human, fully ape, degenerate ape, or a wastebasket taxon. Evolutionists have adapted the story of human origins quite drastically. Have a look at this slide. This linear view of how humans evolved from pre-human ancestors is no longer held to as a valid story. Paleoanthropologists would now agree that the story of human evolution is quite messy. They are forced to reject the notion that human evolution has occurred in a linear fashion. According to the current story of human evolution, there have been countless species, with many of the so-called different species of human occupying the planet at the same time. I mentioned this in the introduction of this statement. According to the evolutionary thinking and storytelling, these many different species, of course, we as biblical creationists would not agree that there have been different species of humans, different variations, sure, have oftentimes shared the same goal and worked towards a shared purpose. Evolutionists would insist this time period, where so many different human species lived at the same time, was instrumental in how modern day humans evolved. Again, have a look at the slide. The tree of life for genus Homo is now depicted as a bush rather than a tree, a bushy mess. Okay, paleoanthropologists have pointed out that the story of human evolution gets more confusing with each new find. We could spend hours just on quotes from their own literature and dialogues and lectures, but let's just go over a few. For example, from Wood here, it says, even with all the fossil evidence and analytical techniques from the past 50 years, a convincing hypothesis for the origin of Homo, notice this, remains elusive. The Pliocene hominin diversity conundrum. Do more fossils mean less clarity? Read the paper. The answer is no. Not any more clear, more confusing. It's a mess. Although there is no doubt that these new fossil discoveries have opened new windows into our evolutionary past, they have also complicated, notice this, complicated our understanding of early hominin taxonomy and phylogenetic relationships. These are not sources and quotes from young Earth creationists. Most hominid fossils, even though they serve as a basis for endless speculation and elaborate storytelling, notice this from Gould himself, are fragments of jaws and scraps of skulls. Oh, that's a quote from 1980. You know, that's outdated. That's a quote mine. Oh, really? Because paleoanthropologists are saying the exact same thing today in 2021. If we want to be objective, we shall almost certainly have to, notice this, guys, have to scrap the iconic list of names in which hominin fossil specimens have historically been trapped. Notice this, and start from the beginning. New hypotheses, new ideas when it comes to relationship. And we can go on and on and on. Look at this. Fossil find of the century may just be a gorilla after all. Study of partial left femurs suggests Sahelanthropus chidensis was not a hominin after all. Others suggest it's just an ancient ape. Again, all new discoveries make things more confusing. What a mess early Homo is. The transition of Homo continues to be totally confusing. As we have seen, the ape to man evolutionary story is a perpetually shifting story. The tree for human evolution is now extremely bushy and convoluted. The field of paleoanthropology is contested and fragmented. The field, as we have seen, is fragmented just like the fossil evidence, the low quality, low confidence fossil evidence. Fossil evidence is scant, it's scattered, and the opinions and understandings of these various scientists in the field are scattered. Just have a look at their lectures, their dialogues, read their technical papers. Everything, every finding is contested. Every finding is debated. But this is where we are supposed to put our faith. Do we put our faith in the opinions and the wisdom of man, or do we put our faith in the and trust in the power of God, the one who is perfect and infallible? Notice this, the separation of man and apes. Paleoanthropologist Leslie Aiello, who served as head of the anthropology department of University College London, stated that when it comes to locomotion, Australopithecines are like apes, and the Homo group are like humans. Something major occurred when Homo evolved, and it wasn't just in the brain. Years ago, Mary Leakey discovered footprints. Notice this on the slide. This is fascinating stuff. That are indistinguishable from modern human footprints. As a matter of fact, if these footprints were discovered today, nobody would question these footprints. They would all conclude they were made by barefooted human beings. Evolutionists have actually dated these footprints to roughly 3 or 4 million years ago. And therefore, because of their circular reasoning, they cannot conclude that they were made by humans. Just have a look at the different variety of primate feet to the right, and then the actual footprint itself with the inline big toe to the left. Now, let's get more into the human evolution icons when it comes to the Homo genus, okay? Human evolution or genetic degeneration. Well, so-called primitive humans are actually inbred subpopulations. We saw this earlier with a few of the papers and technical articles describing Neanderthals as being highly inbred, 40% less fit than modern humans. Genetics now know that the human race is accumulating harmful mutations at an alarming rate. It's roughly 100 new mutations per person per generation. Most of them unselectable, most of them invisible to selection, and most of them effectively neutral. That's why they're invisible. They're not large enough. They're not detrimental enough for selection to see them, for Mother Nature to see them. So what type of selection can Luca here present us with today that can remove so many deleterious mutations that are accumulating from generation to generation? Now, notice this, guys. This degenerative process accelerates whenever a small population is reproductively isolated for many generations. This results in inbreeding and accelerated mutation accumulation. Furthermore, when just a few closely related individuals start a new and isolated population, the result will be founder effects, whereby their descendants will manifest the genetic peculiarities and defects of the founding individuals. And this is exactly what we see for the evolutionary icons such as Neanderthals, Erectus, Hobbit, and Naledi. They're not primitive pre-humans. Okay, Neanderthals lived in the harsh climate of glacial Europe, where only a sparse population would survive by the hunting. Naledi lived among the caves of the South African wilderness. Hobbit lived on the remote Indonesian island of Flores. Each of these settings would lead to founder effects, isolation, and inbreeding. These things would lead to genetic pathologies and anomalous skulls. So they want to pick up these anomalous features and say they're primitive or basal or transitional, when in fact, they are due to pathology, they are due to disease, they are due to genetic degeneration. Notice this: Neanderthal was highly inbred and had a very high genetic load. Hobbit was also inbred, apparently suffered from a special type of genetic degeneration called reductive evolution, results in decreased body size, results in decreased brain size. Naledi, likewise inbred and suffered from reductive evolution, resulting in the same features. Erectus, same thing. Less advanced, but still there. Notice this quote. This is fascinating. It is remarkable that so many features similar to those normally present in great apes, in Australopithecus and Paranthropus, and in early Homo, but not in modern Homo sapiens, are generated in humans by growth deficits due to the absence of thyroid hormone. In other words, notice this is important, guys. Many of the pathological features of cretinism mimic the primitive characters of evolution, making it easy to what? Mistake pathological features for primitive characters. The differences can be disentangled by understanding the underlying biology of characters. So where these evolutionists oftentimes hold up, an anomalous feature and say it's primitive. In fact, we have a paper here by Eric Trinkhaus where he's saying this is what we see in the fossil record when it comes to the late Pleistocene. Is we see skeletal pathologies. We essentially see plenty of evidence for degeneration. So with a minute here, I do want to touch on a couple things: Afarensis and the other Australopithecines. I mean, what we see overall is hands like an ape, feet like an ape, knee joint of an ape, body size, limb proportions of an ape, skull, face, brain size, teeth, jaw of an ape, and just a lot of imagination. 17 minutes really does fly by. So that is my time right there. And I look forward to the rebuttal and the discussion because I've got about 500 other slides to get through. So I appreciate it. And thanks so much.
All right. Wow. Perfect. Right on time. It's like you planned for it or something. All right. We are going to jump into our 10-minute rebuttals. I just wanted to remind the audience, if you could please hit that like button. That really helps the channel. The algorithm boosts these things up if you want to see more debates like this. Again, also promoting John's after show that is going to take place when this ends over on his channel. He's posting the link in the side chat. And we will jump in there right now to the rebuttal to these introductions. So Luca, whenever you are ready, I will get the timer going for 10 minutes and I will begin on your first words.
Okay, I'm ready. So it was quite a good presentation. Sadly, I almost agree with everything. Not like I agree with Donny's points, of course, but I can't say anything about those. I'm not an expert, and if you give me a bone of an ape, Australopithecus, or anything like that, I will be just able to say that's a bone. That's not my field, and I did focus on something else for a reason. But I can say some things. First of all, I cannot agree on the degeneration part. I live in Italy, and that's quite important for this line of evidence. Why? If you look at the very old buildings, I went in Milan some time ago, and I was there in very old houses. And if you look at those, they had very small doors. And when I mean small, I mean small. During the Middle Ages, people were much smaller than me. And also, I'm a teacher. I'm with younger people all the time. And I've seen 10, 11 years old, almost as tall as me. I do not think that people are getting worse by the time. Of course, evolution is not a perfect process. We can talk about that a lot if Donny will want to do so, but not an expert on those topics. even though I know my fair share. Why I want to focus on endogenous retrovirus? Because during my experience, high school experience, I did tinker with those kind of things. My school was a school centered around agriculture and zootechnic. So all about plants and animals. We had some animals too, and we had some laboratories. One of them was a laboratory where we were cloning plants. So I was working with DNA and stuff like that since when I was 17, 18 years old. Of course, it's not like I was doing all that by myself. I was very young, but I have to say that I've studied very, very advanced things. I was very surprised by the level of my instruction, my knowledge on the subject, because I found out about these things very when I was very, very young, and looking about it on the net, I managed to find those topics only in technical papers. So my instruction on the topic was that good, and I was very surprised that something that it was perceived from me as quite simple, it was indeed very advanced. So I can talk about these things because I have direct experience. It's important because when I debate, a lot of people tell me that I'm indoctrinated, that I do believe what I believe, and it's not even a belief. Sorry, because I've seen evidence for it, so it's not like I just believe in something. And I did not ever explain my experience. I did some work with those kind of things. I even run some tests. I did run some tests on a GMO plant with electrophoresis machinery when I was only, I think, 18 years old. I don't know if the people there know what I'm talking about. It's a machinery to analyze the genes inside DNA. It was a test to find out if the plant was indeed a GMO or not. So I was into very technical stuff when I was younger. And then, of course, I started to study chemistry. So I cannot wait to discuss these topics. I think that we will have a pretty good discussion on those. And I do think that RNA endogenous retrovirus are a very good piece of evidence because I cannot bring myself to think a reason to design something like that. So I can stop with that. I leave my time for the discussion or the Q&A.
All right. Awesome. 7 minutes, 30 seconds. Not bad. So only 3 minutes left anyway. So Donny, when you were on mute and when you were ready, I will start that 10 minutes. And thanks, guys, for all the questions. They're pouring in. So I won't probably be able to get too many more. Sorry. Okay.
All right, I appreciate it. Oh, I'm sorry. Go ahead, Matt.
No, it's okay. When you are ready, I'm going to mute myself, and then I'll start the timer when you go.
All right, I got 10 minutes here and a lot of points to address, so I am going to start now and talk as fast as I can. I'll be sweating, I'm sure, by the end of this. So, Luca, I appreciate your opening, and the burden of proof today is on Luca to provide scientific evidence for evolution. I really like Luca. I don't want to be, overly aggressive or mean, but my opening statement did provide some really undeniable evidence showing just how contested and debated and just how fragmented the field of human evolution is. Paleoanthropologists don't really have any good answers. Currently, the fossil evidence, as we have seen throughout the opening statement, is no support for ape to man evolution, and neither is the genetic data. So let's get right into Luca's many points here. So yes, we do share much with the apes in terms of anatomy, physiology, morphology, and genetics. Evolutionists would say non-human apes when it comes to like the chimpanzees, bonobos, gorillas, so on and so forth. Since according to the law of monophily, the evolutionists would say that we are apes. Heck, we are even eukaryotes, right? Does that mean all eukaryotes are related? Of course not. Of course not. Linnaeus and classification systematics, taxonomy, cladistics, for example, I'm sure everybody's heard kingdom, phylum, class, order, family, genus, species. Yes, we can classify animals. I don't think the animals themselves care where we classify them. Classifying various animals based on characteristics and traits and features and genetics does not actually prove relationship, because similarity does not equate relationship. Okay, so let's make that clear. If we were to draw a circle big enough, we can just add about everything into it. How about we draw a circle that says living things? Let's title our circle living things. Just because everything can be grouped into that circle does not mean everything within that circle is related. I'll ask Luca. Luca, do you believe that you and a mosquito are related through common ancestry? Or do you believe that you and a strawberry are related through common ancestry? Okay, just because everything can be grouped into that, say, circle, does not mean everything within that circle is related. We could draw a circle and call it modes of transportation. Massive circle, modes of transportation. Wow. Apparently now all vans of all manufacturers are related to all sedans of every other manufacturer. Homology is not really a good line of evidence for common ancestry. Why? Because similarities can be due to common design. Chevys and Hondas both build their cars with, believe it or not, windshields, doors, a steering wheel, and wow, even tires. Many modes of transportation have 4 tires, and those tires being made out of rubber. Wow, Chevys, Hondas, Mercedes, and Fords must share a common ancestor. Maybe they inherited these features from a common sedan ancestor in the Arctic millions of years ago. Vehicles these days are even designed and built with control features, lane control, adaptive headlights, backup cameras, navigation systems. You'll find vehicles all have airbags in common. Require gas, are used to move from one place to another for functional purposes. There are thousands and thousands and thousands of similarities between all forms of modes of transportation. And it's not because of common ancestry, it's actually because of common design. Human engineers build and design in homologous patterns. This is the best way to do it. This is good design. And our model of biblical creation would suggest that because we're made in the image of God, we should be able to get a sense for how God designed the biological world based on how we design things. And it turns out that we design in homologous patterns. It turns out that we design in nested hierarchical patterns. These lines of evidence, these observations are, unfortunately to the evolutionist, agnostic to the debate. It would be like me pointing to the fact that the sky is blue and saying, well, Luca, the sky is blue, therefore creation is true, evolution is false. No, both models, creation and evolution, can explain the data itself. So now I want to share screen because his other argument had to do with endogenous retroviruses. So let's get into that. A couple visuals though. I'm a visual learner, so here, as you can see, thousands of similarities in all types of modes of transportation. Humans design in homologous patterns, nested hierarchies. The fact that humans share more with the chimpanzee than humans share more with the lizard. Okay, just standing back and looking at the physical features, looking at the phenotype, for example, you can predict that, yes, let's just say we're looking at genetics or hierarchies in genetics. Yeah, of course, we're going to predict or expect more similarities between the human and the chimpanzee than between a human and a mouse. Okay, the evolutionist wants to say, well, we share more with the human than we do with the mouse. And of course, both creatures are mammals, but it's because we share a more distant ancestor. Chimpanzees are our closest cousin. We split more recent than the common ancestor we would have with a mouse. And the differences are time, differences that have accumulated. Okay. But the fact is, what we see is hierarchical patterns in modes of transportation. A sedan, by definition, just stand back and look, a sedan shares more with an SUV than it's going to share with an unpowered vehicle, even like a bicycle, or an engine-powered vehicle that we would group like a plane. Okay, so this is expected. And again, what's interesting too, when it comes to humans and chimpanzees, although there is similarities, even within the similar genetic sequences, guess what we have? Differences in epigenetics, differences when it comes to gene expression. The similar gene sequences, but different patterns of expression. So still differences even in that which is would be considered similar. It's amazing. So endogenous retroviruses was his other argument. So we'll deal with it here, okay? Notice this. Viruses in general, guys. Most evolutionists, they seem to assume that when you hear the word virus, think of something bad. No, the majority of viruses are actually good and beneficial. Beneficial to you, beneficial to the eco. You wouldn't want to live in a world without viruses, okay? Now, here's a fact. You have over 10 trillion cells, and you actually have more viruses and bacteria, for that matter, in and on your bodies than you do cells. Well, why aren't we all dead? I mean, if all those viruses are bad and viruses are just nothing but, evil and negative to the health of living organisms and ecosystems. No, because they're there. They're important. They're designed that way. They're designed to regulate, for one, the number of bacterial species that exists within us. Without the viruses, maybe the bacterial species would just take over and we die. No, it's symbiotic relationships. These viruses are necessary to sustain healthy life processes in the cell. Notice this, BBC.com, why the world needs viruses to function. The vast majority of viruses are not pathogenic to humans, and many play integral roles in propping up ecosystems. Others maintain the health of individual organisms. Sometimes I'll be debating these evolutionists and they'll say, oh, I don't believe what you're saying is true. It's like, listen, this comes from your own secular literature. Okay, endogenous retroviruses, for example. Okay, these viral-like elements that litter the genomes of living organisms. How do retroviruses become endogenous? Just for anybody who wants to know, what are these we're dealing with? Human endogenous retroviruses, HERVs, represent footprints. Notice this. Notice the built-in assumptions, guys. Footprints of previous retroviral infection and have been termed fossil viruses. They are transmitted vertically through the germline. It has to be the germline in order for it to be passed on, and are thus inherited by successive generations. Okay, notice the built-in assumptions, because anybody who's familiar with my many debates and talks on this, the real question is, are these endogenous retroviruses and other classes of retrotransposons, are they really the leftover remnants of past viral infections over millions and millions of years, or are they created units of DNA function? One model, the evolutionary model, would predict that they are evolutionary leftovers, genomic fossils, and therefore junk. We, as creationists, would predict, okay, if they're created units of DNA function, they are put there and front-loaded for a reason, for a purpose. We would predict what? Function. Notice this. Notice this from the technical literature. Look at this, virology. How endogenous retroviruses, believe it or not, protect us from viral infections. Long disregarded as junk DNA or genomic dark matter, endogenous retroviruses have turned out to represent important components of the antiviral immune system. Not only regulate cellular immune activation, but may even directly target invading pathogens. And notice this. Gene expression. They're functional in gene expression, determining cell types, functional in the embryo, functional on so many levels. Okay, the LTRs that he talked about, the long terminal repeat element of an ERV has the capacity to exert regulatory influence as both a promoter and enhancer of cellular genes. And the creationist position has an entire model that talks about these endogenous retroviruses. And for example, notice this. Did you know that cells produce many of the same things that viruses are made of? We make protein codes, we copy DNA and RNA, we have mechanisms of moving DNA to different parts of the genome. Thus, some viruses may have originated in normal cellular operations. The parts are all there. Sometimes the parts are assembled into things that almost appear virus-like. And wow, that's 10 minutes. And these rebuttals fly by, but I think I addressed all the points, so I'm good.
Yeah, they do go quick. They do go real quick, that's for sure. Debating is kind of stressful. But now we're going to jump into the 30-minute discussion portion of this debate. I've already gathered up a lot of questions, so I don't even know if I can probably take more. I will, but we might not get to them, just so you guys know. A reminder that John will be having an after show. He's posting the link on the side. And if you guys wanted to grab a drink real quick, I can go through the upcoming debates that we have on this channel in the next probably week or 2 weeks. The one for tomorrow is going to be a debate on oneness by Kelly Powers versus Rodney Smith. That will be at 6:30 p.m. Pacific Standard Time. And then on 3/28, so in about, a week, almost a week, we have a debate. Is evolution a responsible scientific theory? It looks like that is T-Jump and Kent Hovind at 5 p.m. Pacific Standard Time on the 8th. The next one will be the Great Apes of the Earth debate with Professor David McQueen versus, the name goes off the side, sorry. That will be on the 9th at 3 p.m. Pacific Standard Time. The next debate will be, is evolution a reasonable scientific theory? Again, with Kent Hovind. And I can't make out who he's debating in this one either. That's on the 11th. Man, Kent just debates every day, doesn't he? And then the next one, evolution debate. Again, transitional fossils. This is Christopher Sylvius versus looks like Jackson Rowe in the picture. Name goes off the side. That will be on the 12th at 6 p.m. So a lot of events coming up. Now into the discussion portion, guys. So I will set the timer for 30 minutes. And who starts off on this one?
Okay, maybe me.
Yeah, you would go, Luca, because you went first.
So it was good, especially the last part. But I still think that ERVs are good evidence for evolution. Why? Because when you look at them, and if you remember, we did have that discussion, how we can detect those things and why. It's important because we know where enzymes can cut and how they can cut the DNA. And when you find something like that, a sequence where there was a cut, or at least look like it, and then 3-4 genes for structure useful to a virus, and those things can be useful for us too and make sense, because any mutation can change those proteins, and those proteins can be useful for us. For what I know, we do have a placenta for a things like that, a thing like that. So that makes sense, but on the design standpoint, it does not make any sense. Those sequences, for the most part, are non-coding regions, the LRTs, because those are the regions where the cut was. And it's quite important. And even the inside, it's not always, we can say most of the time, is not useful for us and can be even dangerous in some cases, because those are and they are remnants of a virus. We can trace those virus. We do know, even for quite a lot of them, what kind of virus they were. It's that good. Please.
Well, I appreciate that, Luca. Some good points there worth addressing. And since I love visuals, I'll pull up some visuals here. So again, I would counter that by saying this whole observation of these insertions, even according to the evolutionists, this would have occurred millions of years ago. So we're looking at the remnants, or you could say the genomic fossil evidence today. Okay. Firstly, I'd like to ask you just a real quick question, because I do have a lot to respond to. So if you give a quick answer, did the evolutionary community, and if you don't have an answer to this right now, that's fine. Did the evolutionary community predict that these endogenous retroviruses, or, I mean, I want to point out these DNA elements did not name themselves endogenous retroviruses. Okay. So I don't even think that's a good name for them, but let's just go with it since the evolutionists picked it. Did they predict function for these, and I'm going to call them viral-like elements, even the ALU sequences, or any class of retrotransposon? Did they predict function prior to the discovery of function in ERVs? Quick answer, if you could, Luca.
Okay, I do not know what was predicted. For me, make perfect sense, and I said why.
Okay, I appreciate that quick answer. So here's the thing. It goes back to what I said in my rebuttal. Are they really the ancient remnants of past viral infections? I went over an article there. I could go over a number of papers showing how viruses in general. Okay, now I'm differentiating between the viruses that were being referred to, obviously, in that technical article, and endogenous retroviruses. But I'm just pointing out that the virome in general is necessary. It's necessary to sustain healthy life processes in the cell. It's necessary for regulation of bacterial species, necessary in the ecosystem. And these retroviral-like elements, we as creationists have predicted the same thing. And it just so turns out, and I have a feeling the evolutionary community and the evolutionists that I debate and engage with, I really do believe that they wish this data did not come to light. I really think that they wish this wasn't true. So, for example, here's a summary of an article that openly, explicitly denying that retrotransposons are junk DNA. Far from being junk DNA, and notice this, it's not just a couple. The pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin. A new study demonstrates how the transcription of one such element, HERVH, can modify the higher order 3D structure of chromatin during early primate development. So I want this to be free-flowing and organic. So I want to show you one other paper, and then I'm going to ask you a question, and then you can take your time. So notice these. Retroviral promoters in the human genome. These data suggest that, and I've debated evolutionists where they say, well, it's just one or two, but most of them are not functional. Notice this. These data suggest that ERVs may regulate human transcription on a large scale. Endogenous non-retroviral RNA virus elements evidence a type of antiviral immunity. And I've got a paper here somewhere that goes into embryological development. For example, here. Endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. So this is a confirmed prediction of our model of not only created heterozygosity, but of the variation-inducing genetic element hypothesis that would say these fragments, these DNA elements are beneficial if they're there and we share them with the primates and the biological world. We would say we share them because of common design, and therefore they must be functional there for a reason. So here's a question I have for you, Luca, and then respond to whatever you want. Okay, there's a certain class of retrotransposon. I believe it's found in mice, where while the mouse is developing, if they snip it out, okay, if they knock it out, the mouse stops developing and it dies. And it's because the mouse depends on that specific class of retrotransposon. So my question to you is, how did mammals survive? Let's say, how did these mice survive before apparently they got this insertion, and then eventually co-option occurred where this novel function in the embryo was at? Were mice not having babies? I mean, how did they have babies without this? Go ahead, take your time.
Okay, that's a very good question with a very interesting answer. How did they survive? Well, let's take an example I did, the placenta. You can find organisms without a placenta. If you look at the women in general, when they are pregnant, if you look at the fetus and everything around it, it looks a lot like an egg. We just develop another structure to render the shell and other features useless. So what they were doing before that infection, I don't know exactly, but I can guess they were different organisms with different mechanisms. Like for us, we are ancestor. Let's pretend that's a confirming thing, but it's just for discussion. Our ancestor, they were producing eggs, and then something changed. The eggs maybe lost is not a shell per se, but maybe it was more soft. Then it was no longer a shell, and then the placentas came in to make the connection between the baby and the mother. And it's not like you will ever have something unable to live. Maybe some non-optimal solution time to time, but everything in that progression will be able to survive. On the retrovirus, yes, I'm not saying that's junk. Some of them are, because some of them are not even that deteriorated. We can even tell what kind of virus were, so they are quite preserved, and those are not useful to us, of course. And if we are alive right now, it's because a lot of those elements are just deactivated. If some got reactivated again, you can have diseases, little ones at that. So depends, and makes sense for those elements to do that. Yes, because if you think about it, I have to ask you, did you have a good idea on how DNA produce protein, on the mechanism, more or less?
Yeah, I was just right. I was writing frantically everything you're saying, Lucas. So we'll make our way right to the last thing you said there. So, to me, that does sound like a whole lot of storytelling. Maybe this, maybe that, and as I always say, you can believe that, capital B, believe. But it's not science, because where's the predictions, where's the empirical evidence for this story, where's the empirical evidence demonstrating non-functional endogenous retroviruses going from non-functional to something incredibly functional and necessary, critically functional in the embryo, critically functional in determining cell types, critically functional in regulating genes, in acting as antiviral protectors, and beneficial and functional in the immune. I mean, we can go on and on and on. Those are just the ones that come to mind. But when it comes to what you said, some animals have placenta, some don't. Sure. There's great diversity in the biological world. I mean, you can find modes of transportation where some have a diesel engine and others have a regular engine, or some are all-wheel drive, others have front-wheel drive or rear-wheel drive. I mean, there's all sorts of variation in the biological world. And what I find interesting is we do have taxonomically restricted ERVs that are functional, specific to humans, right? And then we also share many others with the chimpanzees, but we don't share an identical number with them because we are different and we have different functions. Although we do share the most with the chimpanzee than any other form of life, right? We share more with the chimpanzee than we do with a dog or a fish. So we'd expect some degree of similarity there. But I think if there's any validity to what you're saying, Luca, you would have to show how these functional components can be added to the endogenous retrovirus. Because the last thing I'll say here is, if a blindfolded painter, let's say he had a painting and then he put a blindfold on and he just started, adding random brush strokes to it. Well, he's more likely going to mess up the painting than make it any better than what it was. And that's what we see with the endogenous retroviruses, or these various classes of retrotransposons, is since they're functional, but the evolutionists are saying, well, these functions were co-opted or adopted at some point. It's like, wait a minute, this is a viral infection. This is an insertion. It's more likely to corrupt genetic functionality and genetic information than actually make it better, to adopt a function in the embryo or the immune system. I understand the evolutionists have, a story, or what I would say a rescue device, but I just want to see the empirical data. I want to see a technical paper demonstrating that this is possible. Yes, we do have a hypothesis that says a lot of these viruses originated from healthy viral-like elements, because the genome has all the different, I showed a slide, Luke, I don't know if you remember, the genome has all the different components to accidentally make a virus that may be harmful. And then a virus that is harmful, or let's say a virus that's in its host and it's healthy in that host, regulates the various functions in that host environment, but then it jumps to another species. That's where it can become bad. That's where we see a lot of these bad viruses, because it is a virus that has jumped from one species where it was good to another species where it was bad. But Luca, here's the last thing I'll say is retroviruses need a host to replicate, right? So what came first, the host or the virus? I said a lot there, so go ahead, take your time.
Okay. So that's quite a complex thing, but I will try. When a piece of DNA is creating a protein, it's like a factory. You have those strands of RNA that binds in a certain location of the DNA in a certain order. Those strands bring pieces of the protein, an amino acid. When those things connect with the DNA, they, like a factory, will build the protein step by step. When you start with the viral infection, you have those proteins building things for the virus. and that's harmful for the cell. Okay, and if things remains like that, the cell will die, of course. But time to time, you can have a mutation. Sorry, there is some kind of noise. Okay.
I muted. No
Uh,
worries.
okay. So you have a mutation, and then the protein has no longer any function. It's useless. If things remains like that, the portion will be ruled out sooner or later, because a cell can produce something that's not harmful but does consume energy to do so. If it's useful, it will be kept. If not, it will be rolled out. Time to time, it can you can have a good mutation with a result that's good for the cell, a slightly different protein that can be useful to that cell, to that organism. So it makes perfect sense for things built to reproduce proteins for a virus to produce something good, even for us. Of course, not every retrovirus does that, but it's a
Well, Luca, if I could interject one thing,
yes.
and I understand what you're saying, and we could go, down the beneficial mutation route. I definitely want to get to a question to you pertaining to something in my presentation, but we don't have to rush there. I just want to point out, I don't want to discuss ERVs the entire time, but again, I want to see empirical evidence. I understand the evolutionists have a story as to how co-option could occur. Today, what we see is the remnants of these retroviral-like elements. And what we also see is very important functions associated with them. All I'm asking for as supporting evidence for the story associated with how these ERVs co-opted these various functions is something today, maybe in a lab, a technical paper that actually shows a non-functional endogenous retrovirus going from non-functional to something incredibly and critically functional in, let's just say, the embryo. That's really all I'm asking for.
Well, we do have lizards developing a placenta, so those kind of process are observed right
Well,
now.
I mean, like specifically the ERV though, because we're talking
Yes,
about
it's not like I can bring you a specific paper on that, but I can search you if you want. I know that the placenta in mammals is from a process like that. We do know the kind of virus, we do know the kind of mutation that went in that place. So evidence is there, and the process I was describing, it's an actual process. Those happen, and we can observe them. It's not like it's rocket science. It's the process to make a protein. It's a well understood
right.
process. My point with ERVs, it's their design. You have two non-coding regions, the LRVs, then four genes, for the most part, remnants of viral genes. It's how we know those are remnants of an infection. And of course,
Well,
you
but
can
here's
have
the thing: these
mutation like
components.
in every point of your DNA, and that's now part of your DNA. So you can have mutation even there that can change things and can be useful or not.
Yeah, but these
You can
components,
get
Luca,
sick.
are functional. Like I showed the slide from a paper showing that these LTRs are helpful as in terms of promoters and regulation and but at this point let's
Yes.
leave the ERVs up to the audience, because before time runs out, Luke, I do want to ask you, because here's the thing. Your burden of proof tonight is there scientific evidence for evolution. I don't think any evolutionist has adequately answered why we see functional ERVs, but you know what, you made your case. Maybe we can leave it up to the audience. I am curious though, from your perspective, from your model, Luca. I showed many slides and many data points, including quotes from paleoanthropologists. I guess, firstly, are you familiar with the Leotoli Footprints?
Yes.
But, okay, so if you are, how do we have anatomically modern-looking human footprints back, according to the evolutionary model, 3 to 4 million years ago?
Okay. I did my fair share of search on the topic, and I've looked into it, and those, even if they are similar to our footprints, they have a quite different way to distribute the weight. The toe is quite different from our toe, so those are not humans. Those are not from a human as we.
Well, are you saying because of the way that they walk?
No, it's when you study a footprint, it's not that easy. You cannot just look at it.
Right.
I've seen studies on those prints, and they are different from ours.
Well, no.
There are differences, quite important.
I know what paper you're talking about. But here's the thing, where they say, well, there's a different weight distribution, or firstly, for the audience sake, look at the visual. You got the inline big toe. I made it easy here. We got a bunch of comparisons: gorilla, chimpanzee, okay, orangutan, baboon, human. Inline big toe. It matches up nearly identical. Now the evolutionist wants to say, well, it's an intermediate stride, or the way it bears its weight is transitional because they did some comparisons and said, well, it's slightly off from the way humans walk. But that's ridiculous, because, and I'll say this, Luca, and give your thoughts. If I were to make footprints in a sand, let's say I were to go walk through sand on a beach. When I first get up, have a coffee, I'm fresh, I'm walking nicely. Then I go to work for 12 hours and I've been on my feet all day and I'm tired, I'm exhausted, my feet are sore. And then I go do the same thing. Okay, walk through the sand, and then they test. Okay, they do their analyses. They're going to conclude, wow, these are two different, people or creatures walking, because there's different weight distribution, there's a variation, and so on and so forth. Or what about somebody who runs a marathon? Look at the way they run, look at their strides, look at the way they're performing at the start of the marathon versus after. I mean, there's variations in the way we just as a person walk throughout the day. That's a weak argument. I've looked through that. Guts and Gibbon brought that up. I don't find that. I think that's a rescue device. But yeah, go ahead, Luca. What do you think about that?
No, I do not agree. Of course, I'm not an expert, so my thoughts are good as yours. But if you look at the image, you can see that there is something strange in the toe. So you don't need to compare this to a chimpanzee or a bonobo or things like that. You need to compare those footprints to a foot of like to Lucy. Lucy, of course, does not have the foots.
Right.
There are other Australopithecus with that.
Yeah, so and it doesn't look like that though.
Idea of how their feet were, and they were slightly different from ours, and that footprints look like it. But I'm not an expert. It's just a layman giving an opinion on that. And I do not have that paper here right now, so it's not like I can discuss anything.
Right. No, that's fine. Okay, well, based on what you're saying, let me ask you this then, because when they do actually find evidence for Australopithecus-like feet, they see it's not like the Leotoli footprints, unless, for example, they've admitted, like Lee Berger himself has admitted, that Afarensis is probably a chimeric species. This was their original conclusion anyways, because we know, as you can see here from Donald Johanson himself, the collection suggests that Homo and Australopithecus coexisted as early as 3 million years ago. So I'd be curious as to your thoughts on this, okay? Is we understand that domestic dogs ultimately go back to wolves. Okay, so obviously domestic dogs today are younger than wolves, but yet still exist. So it'd be like somebody saying, well, if domestic dogs came from wolves, why are there still wolves? Well, that's just misunderstanding of evolution. But if the domestic dogs were before the wolf, like for example, I can live at the same time with my mother or my grandmother, but I can't coexist with my great grandmother. That can't happen. So why do we see such extensive coexistence with the Homo genus and the Australopithecines? We find this author. I mean, here's a relatively new paper that even just a couple million years ago, we have Australopithecus, Paranthropus, and the earliest known Homo erectus, which we would say is fully human. I mean, I've got sources here going over how Erectus was a sailor. They had evidence for purposeful navigation. And a lot of their anomalous features could be attributed to degeneration, just given the environments and conditions that they lived in. Yeah. So how do we have such extensive coexistence and overlap? And then also, how is that evidence for ape to man evolution? And then let's say not the biblical creation model, which we would say, yeah, that's what we would expect: overlap and coexistence. Luca, I said a lot there, so just take as much time as you need.
Don't worry about it. Again, not my field at all, but I did follow a lot of these debates. I love them, of course. So, what do you mean by chimeric? Because when I see Lucy's bones, and not an expert, again, just a layman in that field, but when I see her, I see bones that are like ours, but they are not the same. The pelvic bone, much like ours, but it's a little bit different. He was able to walk upright, but not as good as us. It's a very interesting fossil because their skull is very different from ours. Their body, no, it's almost like ours. Of course, fingers, they were curved. It's not like we were the same. But if you look at Lucy's skeleton without any expertise, you will think that's human. It's much like humans. You need to be an expert to see the difference. Just look at it. The skull, completely different, but the body, not so much. Though there are differences, and those are very interesting ones. So what do you mean by chimeric? Because the single bones are in between us and apes.
That's a good question. So firstly, Lucy here, as you can see, hundreds of pieces. This is how Lucy was found, Afarensis. Hundreds of pieces scattered across a 3-meter area of a hillside. Okay. And if you were to consider everything, including the hand and feet bones, only about 20% of Lucy was found. I mean, for example, they find this. This is what they make. You know, you'll hear a lot of evolutionists. They'll say, you know, you hear from Gutsy Gibbon, there's 400 specimens of Afarensis. We have great evidence. What they actually mean by specimen is this. the single bones that they have. But yet in textbooks or in videos, you'll see like 400 full skeletons of Afarensis, walking around. It's like, no, we're looking at bone fragments. We're looking at 400 specimens, as in 400 different bones. They're scattered, they're disarticulated, they're fragmented. But we also know, and I provided various quotes from these paleoanthropologists, including Donald Johanson himself, Mary Leakey, the Leakeys, Elabin, Yoel Rack. I mean, they all agree that what we're looking at is coexistence between humans and Lucy. But then you say, oh, the features are intermediate. Let's say the ones that are ape, and then the ones that are human. I mean, Lee Berger, take it up with him, just in a recent video that he put up. He said Afarensis is probably chimeric, a mixture of different bones. This isn't a conspiracy. It's just well known. But you'll notice this. The fingers of Lucy's kind were curved for tree life. And she had these, I guess I don't have the slide right here. She had inner ear canals that were better suited for walking on all fours rather than having the balance capabilities of walking like a human. Now, it might have had a different, the Australopithecines probably have a slightly different gait and way of locomotion than like the living great apes, but still nothing like the human great apes. And overall, I put a slide here just to make it easy. No, most of what we see is the fact that body size, limb proportions of an ape. I mean, Lucy's like what, 3.5 feet tall, skull, face, brain size, teeth and jaw of an ape, brain size, inner ear canals. This is what I pointed out. Shoulders, rib cage, spine of an ape, hip of an ape, hands, feet. We don't even really know what the feet look like, but we have the Leotoli footprints, which were found miles and miles and miles away, I think, and even in a totally different layer that they want to attribute to either Afarensis or Animensis, maybe Africanus, when in fact, I would assert that they look like modern, anatomically modern, human-looking footprints. So yeah, when you find these, when you find all these bones in these bone beds and they're disarticulated, they're fragmented, that's what happens. I mean, it took 40 years for them to realize that I believe one of Lucy's vertebrae was actually from a baboon. And so for 40 years, they had that bone being showcased as one of Lucy's bones, when in fact it belonged to a baboon. And last thing I'll say, and then take as much time as you need, Luca, is see, here's the thing. You have two different types of paleoanthropologists, okay? You have what's called those that want to focus on the arboreal related features that they would like to point to and say, see, this was more of a tree dwelling. Plus, there's evidence that Lucy died falling from the tree, out of a tree, but based on the fractures and the evidence there. But they'll focus on these bones and say, see, it's more consistent with like a tree dwelling creature. And then you have the terrestrials, where they'll look to different bones and say, and emphasize those and say, no, see, it was mostly an upright walking creature, when in fact, given the chimeric nature of Afarensis, you have some paleoanthropologists. This is why there's contradictions in debate, because you have some paleoanthropologists that are looking at bones and holding it up and saying, see, this is more human-like, when in fact, no, it is a human bone, because we know the Homogenous and the Australopithecines coexisted and intermingled at the exact same time. I have a quote, I think it's from Leakey, where they said that humans probably ate robust Australopithecines. No wonder why we find them together. So yeah, I said a lot there. Most of it is ape-like. And then the ones that look more human, since they're disarticulated and fragmented, there's even people in the field of paleoanthropology that would admit, yeah, we've got some human bones mixed in with Australopithecus bones. So yeah, go ahead, Luca. What are your thoughts on
that? I have some things to say. First of all, one of those images with the ones with all the bones, it's cut. The complete photo show a bunch of skulls at the end of the table, so it's not complete, and it's important to say so. And it's not the first time that I saw that photo cut like that. The one at the Creation Museum, Lucy, the reconstruction is wrong, and I can tell so, even though I do know nothing about bones and things like that. If you look at Lucy's pelvis, there is no way that's an ape walking on four legs, because the pelvic bone is totally different. Look, chimpanzees, one and human and Lucy is almost identical to us. There is no way that's like a chimp. No way. And I do want the people there to look at it, because it's totally different, really. So I think that those things are quite important. We do have a lot of fossils. A lot are small bone fragment, and does make sense. Very little of what dies became a fossil. So we did find a lot, if you think about it.
Coming up on time.
Sorry?
I said we're coming up on time.
Okay.
I guess since Luca started it off and then he gets a closing statement, let me get the last word here and then we'll hand it to Luca for his closing. Okay, so the last thing I'll say is, nobody said that although I do believe that the Australopithecines are a lot more ape-like when compared to apes today, extant apes, than evolutionists want to believe, since we do have what I believe is a clear mix of human bones and ape bones and all other types of creatures as well. But overall, what we're looking at, they're not identical to what extant apes look like today. My mouse froze there real quick. But notice this. They're around 3 to 4 feet tall with long arms and short legs, ape hands and feet with long curved fingers and toes, opposable thumb-like toes, ape size and ape-shaped skulls, funnel-shaped rib cages with high-placed shoulders for suspensory behavior. Yet they also exhibit, okay, so notice this. This is why they're not exactly the same as the great apes today. They exhibit their own unique morphology. Right? We had a great diversity of apes in the past, great diversity of apes today. As a matter of fact, life was more diverse in the past. That's what we see based on the fossil record. So they had their own unique morphology. The Australopithecines are best described as an extinct ape genus. In the past, there appears to have been a greater diversity of apes than are alive today. The living great apes include Pan, Gorilla, and Pongo. If they were not extinct, so let's say the Australopithecines were not extinct and they were living today among the living great apes. Well, guess what? The Australopithecines would be considered a fourth hominid genus. All four genera share many similarities, and all four are unmistakably ape, yet each one has their own unique set of features, which makes them different variations or species, right? That merit their separate taxonomic designations. So yeah, the fact that these Australopithecines have some variations compared to humans and the great apes, which evolutionists would just say are non-human great apes, doesn't demonstrate any form of relationship to humans. It's kind of just what's expected, is that there would be some differences. But you know what? I'll end it there, and then Luca gets his closing statement. So
great. Yep, 5 minutes whenever you're ready, Luca.
Okay. No, I'm ready. So my points to me still stands. Airways makes no sense when you think about it. If you want to believe in a designer for the genomic parts we are talking about, they are composed by 2 non-coding regions at the end, at the beginning, and inside you find broken genes. Sometimes are useful, of course, but you can relate those genes inside to a virus. Why LRVs can be useful? Yes, they can. They make the synthesis of a protein stop. It's a stop. They are non-coding regions. They can be useful too, but it makes no sense to put those there. Those are sites where the genome was cut, or they look like it. If they were designed, it's a suboptimal design by a long shot. It's like you can find scars in the genome, and we can trace back those wounds to our ancestors, and we do share a lot of them with other animals, apes, gorillas, so on and so forth, and you can go back quite a lot and trace our relationship to other order of animals. So that's a very powerful evidence for me, because again, it makes no sense. If you need to do things like that, and you are a designer, you can do it in much better ways than use broken genes from a virus or something that look like it. If a designer designed that genome in that way, it's tricking us, because we can trace even the kind of viruses we are talking about. Why use those sequences in that order with those remnants of a wounded genome? It makes no sense. And for me, the only believable reason to have those is that's proof for ancient viral infection in our ancestor. So I'll stop there.
All right. No problem. And 3.5 minutes. Donny, whenever you're ready. Or is this Q&A? Did you use yours?
Not yet.
Okay. All right, good.
Let me just get my timer. So I got 5 minutes.
Yeah.
All right, I want to wrap.
Want a warning at 1 minute left or no?
Yeah, you know what? Yeah, that would help. Just
Okay.
because these fly by. And I want to make sure I'm hitting every last point. So
All right.
Here we go. Let me share screen, and 5 minutes starting now. So great debate. I really enjoyed that. Very cordial. And as I like to say, sophisticated. It really was some great arguments and discussion. But the main arguments that I heard, especially arguments from classification systematics, including homology, nested hierarchies, and ERVs. I don't think they withstand scrutiny. And again, I've been asking now for many debates, many years, for actual empirical evidence as to how these ERVs can gain this function if they weren't actually built in with that function. Our model, the design diversity model, would suggest that God created the original created kinds, including Adam and Eve, with pre-existing and beneficial genetic variation. And this could be called upon and acted upon through various means. Epigenetics plays a role, of course, because our environment oftentimes leads to adaptation. But God also front-loaded various other classes of DNA elements, such as these ERVs. And obviously, we're littered with viruses. And that's why we, from the common design perspective, we would predict function. And yet this is what we find. Okay, in the past, notice this, vestigial organs. This argument's dead. You rarely see evolutionists use it. There used to be roughly 180 vestigial organs in the human, known in humans, thought to be vestigial. Appendix, tonsils, the thymus, so on and so forth. Now we know there's functions for all of them. Okay, they're not evidence for evolution. Now the evolutionists are taking the vestigial organ argument, they're applying it to genetics, especially since we only know so little about the DNA language. It would be like me going to Russia, as a proofreader for their books. I don't know anything about Russian. How am I going to be able to tell you what is and what isn't a mistake? So already what the evolution is, what Luca is saying here in his closing statement may be, a mistake and therefore evidence for evolution. You don't know it's a mistake. Give it back just 50 years ago, we had this idea, one gene, one protein, one function. Now we know of protein moonlighting, where one single protein has multiple functions. We understand third position codon variation. It's not just there to tolerate error. It's actually involved in information flow. We have secondary functions, multifunctionality. Okay, now we know that these so-called shared genetic mistakes like pseudogenes, guess what? That were once labeled junk DNA are necessary for the cell. And there's paper after paper here going just how functional and important these pseudogenes are. And the same thing goes for the endogenous retroviruses. Okay, notice this. Certain mobile retrovirus particles originally could have moved from site to site within the genome. Okay, this isn't evidence for common ancestry, thereby changing the way genes are regulated within the host genome. And notice this. This is something that is very problematic for evolutionists. It makes much more sense if retroviruses were originally designed as part of the host genome and escaped from the cell after the accumulation of mutations. What's the most parsimonious explanation? It's that they were created. Okay, we have created units of DNA function. The chromosome 2 fusion as well. If you'll notice here, the purported fusion site is actually a functional DNA element inside a human gene. All of their best arguments, pseudogenes, okay, this idea of shared genetic mistakes, they've been overturned. And we went over a number of quotes from paleoanthropologists demonstrating that the field's in a mess. Okay, and we could go over Homo habilis. We can go over every single one of their icons. And the conclusions we make as creationists are conclusions that various paleoanthropologists have made. Look at this. Tattersall has critiqued the entire species of Homo habilis, claiming it is a wastebasket taxon, a rather heterogeneous assemblage, and is probably more than just one hominid species is represented. We can go into Sediba, we can go into Ardipithecus, we touched on Afarensis, and again, we understand that the so-called representatives of the Homo genus, like Neanderthals, Floresiensis, Naledi, Erectus, Heidelbergensis, we understand that given their environmental conditions, given the fact that they were isolated, given the fact that they were highly inbred, okay, you have these mutations that are hidden, they're manifested. And eventually these features, the anomalous features, become fixed. And these anomalous features are not evidence for human evolution. They're evidence for genetic degeneration consistent with biblical creation. And I yield. That's 5 minutes.
Perfect. Wow. Like you debated before. Anyway, good job, guys. Yeah, I got a lot of questions. So we're going to move into that fairly quickly. Luca, how much time do you have left? Because the vast majority of these questions are for you.
Don't worry about it.
Okay.
I can do it not all night because I will die sooner or later, but I will try.
That's all right. We have that after show as well. So I just wanted to plug that again as well. John, when you're ready, post that in the comment section and we will put it up for everyone to see. And we have 2 super chats. One of them is a question, but I will start with the statement. Thank you, Bubblegum, for the super chat. It says, we're out here finding dinosaurs with soft tissue, but not a single transitional of a half-man, half-ape fossil, despite dinosaurs being way older, according to evolutionists. And then we come down to another super chat from Born 100 Years Late. Thank you for the super chat. This question is for you, Luca, and it's regarding the trackways that you guys were talking about earlier. It says, today we saw a rare physical evidence that it is clearly human. A simple search shows people with similar feet today. Why is this ignored?
It's not. I think that study I was referring to, it's a good one. But of course, I'm a layman on the subject, so I can go just that far. I can tell that footprint is off. There is something off in it. If you look at it, the big toe is in a very strange position, if you ask me. But again, not an expert. I yield to Donny.
Let me address that or give my response, Matt. These Leotoli footprints, okay, they are an unsolved mystery for those who hold to deep time in ape to man evolution. These footprints, and I'll screen share them again. We discussed it in the debate. I've been using these footprints now for a couple of years during debates, and all I see is rescue devices. These footprints look nearly identical to modern human. Where's the evidence for humans, living around the time of our so-called Australopithecus ancestors? And it's like we show them evidence of human bones, evidence of human footprints, human-made tools, human-made shelters, and it's like, well, no, that's not evidence. It's like, why? Well, because we know humans didn't live back then. It's circular. It's like the orphan genes. Well, how do you explain the existence of these taxonomically restricted and highly essential genes? Well, de novo gene synthesis. Well, why de novo gene synthesis? Well, because orphan genes. It's just circular argument after circular argument. They can't admit that these are human footprints because they don't believe, according to the evolutionary story, that humans existed back then. Let me screen share for everybody. Just visually, objectively, ask yourself, according to the evolutionist model, 3 to 4 million years ago, these footprints, okay, which I understand, I could be corrected on this. I understand measurement wise, this would be equivalent to like somebody today wearing a size 10 or 11 shoe. And yet these Australopithecines were, 3 to 4 feet tall. Not even sexual dimorphism can account. The type of sexual dimorphism that they invoke for the Australopithecines is like sexual dimorphism that we've never actually. It's an unrealistic amount of sexual dimorphism. Notice this again: primate feet, chimpanzee, gorilla, orangutan, baboon. Okay, and then we got the human footprint with the inline big toe, designed and built for upright walking and running marathons. These Australopithecines weren't running. At best, they had a slightly different way of locomotion, slightly different gait, but nothing like humans today, okay? And here's the Leotoli footprint. Match it up. Only the evolution, only those that hold a deep time have to say, well, that's not a human footprint because humans didn't live back then. I'm sorry, that's circular reasoning. I yield.
Okay, that's my turn, right?
Yeah, you get the last word.
When you leave some footprints, it's not like you will leave an exact print of your foot. It's more complex than that. So those footprints can be bigger than the foot of the actual creature. So it's not that simple, and I will not pretend to understand anything on the subject. I yield.
All right. This next one, you kind of were on this a little bit earlier, but we'll just ask it real quick because someone asked, why does Lucy, a conglomeration, appears to include fully formed human sacrum? If you believe it's a transitional, how so?
Sacrum, it's the I do not know anything about it, but Lucy has not the same bones as us. There are differences. There are on the body. It's quite human-like if you look at it, and you are not an expert, of course. It's not like an expert cannot tell the difference. They can quite well, but the body quite human-like. So, but it's not the same. There are differences, but you need to ask to someone that understands something on the topic. So, not me.
Donny, any comment?
Sure. Yeah. So the sacrum is attached to the pelvis, but again, okay, this idea that afferensis is chimeric, Okay, a jumble of species is a view held to many paleo. Now, this is a view that's typically not emphasized in textbooks and things like that because they want to preserve Lucy as this great example of a transitional form. Okay, notice this. The field of paleoanthropology largely reject what was obvious to the founders of the field, which was that humans beings coexisted with all the Australopith types. They admit this. You find this all throughout their literature. The Leakey team and the Johansson team were consistently finding Australopith bones in the same strata. Notice this, in the same strata as human bones, along with human tools, human shelters, and human footprints. But as we've seen, the evolutionists can't conclude that these are human footprints. And apparently they can't conclude, like, for example, habilis. Clearly a wastebasket taxon made up of some Australopithecus bones and Homo erectus bones. And yet they want to call habilis the handyman, right? But given the features that are ape-like or non-human ape-like is what the evolutionists would say, okay, they could not have made tools. They could not have built shelters. But since we find the tools with them, and yet there's some in the field, and I think it's pretty obvious that it's a mixed, a chimeric species, it's Erectus that was building shelters, building tools, using the tools. And humans, as disgusting as it is to think about, they've eaten all sorts of things throughout history, especially during those times, the rekindling of culture, right? We have, for example, according to our model, the post-flood world and the post-flood ice age and the dispersal after Babel, we would have stress. We would have survival would be key. I mean, like I pointed out with these Neanderthals, I mean, I don't think there's any Neanderthal bone that's found that doesn't have evidence for, or I shouldn't say none, but the fossils that are found with Neanderthal reveal that they had a lot of fractures because it was a harsh, cruel world, and people lost everything, and then they had to rebuild culture, and it was kind of like, like a mini Stone Age. And here's the thing. You'd have Erectus that is building these tools, building shelters. And I've got a quote, I believe it's from Leakey, where they said, humans probably ate robust Australopithecines. Humans have eaten all sorts of creatures just to survive. And these evolutionists have this straw man idea, like, Standing for Truth is saying that an ape is here and a human's here, and they're running at each other at 20 kilometers an hour, and they bang into each other and their bones get jumbled. No, if you even spent a little bit of time reading the literature on this, the paleoanthropological community don't even really know why we find so many mixed bone beds. And they have hypotheses. Maybe carnivores, various carnivore animals ate apes, could have attacked humans, killed them, brought the bones back to caves, things like that. Or humans ate Australopithecus-like creatures. We find their bones, obviously, where the humans would have lived and have taken shelter. I mean, there's various hypotheses even within the paleo literature. Okay, so this is not something Young Earth creationists are making up. This is a real thing, and I'll yield there.
Okay.
Luca again gets the last word.
Okay, I'm not nearly as knowledgeable on the subject as I should be, so I think that those discussions are for other people more in the field than me. So let's stop that. Stop there about this question.
No problem. We'll jump right into the next question. I guess I will ask you 2 questions for every one for Donny because you have so many more. This next one, more of a personal view, I would guess. So no right or wrong answer. Do you think that Africans are lower down on the evolutionary chain compared to Caucasians according to this worldview of evolution?
That's a funny thing because if you look at the evolutionary view, because it's quite a funny thing, the Africans are the real humans. We are the ones interbred with Neanderthals and things like that. So they are the pure ones. So no, evolution, it's not about racism. racism. It does nothing on that field. We whole creature are evolved more or less at the same degree. They just change where they got. Maybe someone is more strong, someone is able to survive without his head, like a cockroach. So evolution says nothing about it. And I do remember the bit about race because I did read the comments live when it came.
Good. All right.
I'll give my response, Matt. So it is the Bible that predicted we are all of one people, one race, the human race. And genetics has confirmed this as well. There's not different races. There's not different species of humans. Actually, genetics has revealed, you'll notice this, human genetic diversity, we have extremely low genetic diversity. We are all 99.99999% the same. And a question I usually ask, and I didn't ask it during this debate because we didn't have enough time, is how do the evolutionists explain the fact that humans have incredibly low genetic variation? If we've been evolving for millions and millions of years, from the Australopithecines up to Homo habilis, up to Homo erectus. Homo erectus exists for millions of years. And some evolutionists would propose that an early variation of erectus left Africa, and out of Africa, that erectus evolved into Heidelbergensis. And then you also have Neanderthals, you'd have Naledi, Floresiensis, probably a more degenerated erectus based on island dwarfism. Okay, so you have all of this over millions of years, all of this change. Well, we know mutations accumulate every generation, and a mutation adds diversity because a mutation is adding something that was not previously there. Now, I'm not saying this is beneficial diversity. This is negative diversity. This is the type of diversity that you get on your car when you park too close to someone else's car at the grocery store and they swing open their door and hit you and you get a dent, or the rust spots that build up. That's the type of variation that we typically get from mutations. But the point is, after millions and millions and millions of years of evolution, from Australopithecine-like ancestors to eventually Homo sapiens, that's millions of years of mutations being added to the gene pool. So humans today should actually have high levels of genetic diversity. But yet we find that humans have low genetic diversity. And guess what? That is exactly what the Bible predicted, because the Bible says that God created two human beings, Adam and Eve, just a few thousand years ago. And right off the bat, that restricts genetic diversity. And what we find is low diversity in humans overall, low diversity in the Y chromosome, low diversity in the mitochondrial DNA. The mitochondrial DNA, when you line up these sequences to the chimpanzee mitochondrial DNA, they're nothing alike. The Y chromosome is only about 35% the same to the chimpanzee Y chromosome when you consider overall architecture, gene content, size differences, and yet the Y chromosome should be relatively similar because it's non-recombining DNA. It's not as messy as the autosomal DNA, where you inherit two copies and you experience recombination. Okay, so that's incredibly different. And we can even we can make a reflection or representation of worldwide diversity in the mitochondrial DNA, the Y chromosome. And what we get is exactly what we would expect based on the biblical creation model of ancestry. Notice this is a mitochondrial DNA phylogeny. We've got explosive growth from a center point. We've only got a few DNA differences separating any two people. The African branch is a little bit longer, okay? But at the same time, overall, humans are pretty well the same. In the Y chromosome, every single male Y chromosome nearly identical. You could take the Y chromosomes and build a family tree as well, and it takes us back to just one Y chromosomal ancestor. Listen, this isn't tens of thousands of mutations here. This is not 100 to 200,000 years worth of mutations. This is only a few thousand years. Evolutionists can't explain that. There's one race, the human race, and genetics has confirmed that. So I yield.
Okay, I just say that first of all, for what we know, there was a bottleneck, and it was quite before what we think about Noah's flood and things like that. But I'm surprised that we made that far, because if you think about humans without any technology, I think that a lion is far better at killing than us. So it's even a miracle that we are here now. So I'm not surprised about the low genetic differences between humans, but it's impressive that you can plot the genetic differences and to basically find out a line to Addis Ababa in Africa. Basically, we can trace our origins to Africa, for what we can tell from the data. Not an expert on it. I think that just this topic will need a debate on its own.
All right. Next question for Standing for Truth from Paleologos. Why do you think speciation occurs in other created kinds, but not humans? Part 2 of that is, if you are willing to accept a different species of Paranthropus and Australopithecus, why not Homo?
That's a good question. So this goes back to the low genetic diversity issue as well. Okay, what our starting point is the Bible, and we make retrodictions and predictions. And what does the Bible say? God created two people, Adam and Eve. That restricts genetic diversity, and that also restricts the ability for change, for quote unquote speciation. God created a world full of animals. Animals created in fully working and functioning ecosystems. This also goes for plants. That's why we see amazing diversity and variation in plants. Okay, we also see amazing variation when it comes to animals. But humans started off as two, and that restricts levels of genetic diversity. Okay, and even when it comes to the word speciation, the species is kind of loaded, because where an evolutionist could say, Canis lupus, Canis domesticus, Canis coyotes are different species. We also know that they all interbreed. Same with tigers and lions. So where they would say different species, we would say variations of the same kind. I mean, they can't even agree on a definition of species. I've got slides here. For sake of time, I won't find them. But I'm sure Luca would agree. They have 50 plus definition of species. Look at Neanderthals. We literally have the genes of Neanderthals. And yet I've had 3 formal debates with militant evolutionists that want to say that Neanderthals and humans and Homo sapiens are not the same species. Full debates. I was just watching a lecture. I sent it to Matt. I've been watching a ton of secular lectures on human evolution from 4 months ago. And the speaker, he literally said that there's no debate. There's no question. We are Neanderthals. Neanderthals are us. Neanderthals are us. We're the same species. We interbred with them. We have their genes. We're really not that different. Now, they were the most inbred people group who ever lived on the planet. Okay. So where some creationists want to say, well, there's different species of humans. Well, let's look on a molecular level. Now, we don't have the genetics of all these different variations of humans, but I will predict. Here's a testable prediction. Based on my model of post-Babel dispersal and migration of people groups, we have some subpopulations isolating themselves on us. Why do you think we find the same anomalous features for the most part on Luzonensis with Homo luzonensis, Homo floresiensis, the Hobbit, and Naledi? Because guess what? And I showed that quote earlier, this being admitted by a non-young Earth creationist, that a lot of these pathologies and fixed anomalies means the whole population has this anomalous feature. Well, guess what? That can happen through founder effects, genetic degeneration, inbreeding, isolation, and result in these anomalous features. And these isolated populations, we find that they have these same anomalous features. So on a genetic level, I am predicting and I'll have it in print in my next book. This is a testable prediction. If we do get the genetics or DNA of Floresiensis, Naledi, Erectus, let's say we get the genetic data from Luzonensis, I'm going to predict we're going to see something similar to what we see in the Neanderthal DNA. We're going to see evidence for inbreeding, evidence for genetic degeneration. We'll find massive rungs of homozygosity. This is what we're going to find consistent with the model that I'm putting forth. Okay, these aren't different species. To say these are different species of humans doesn't make any sense. I mean, these variations come about through genetic degeneration, rapid genetic degeneration. And here's the last thing I'll say. Even when it comes to baraminology, the best way to determine ancestry is still through genetics. A fossil, a bone is not inherited sperm and egg. Okay? Genes and traits are. And here's the last thing I want to say. There's something called homoplasy, where even those in the paleoanthropological community, when it comes to human evolution, they struggle because what's the result of ancestry and what's the result of like convergent evolution? I mean, look at this example here. We have sharks, ichthyosaur, and dolphin. Okay, streamlined bodies and fins provide a big advantage for them, allowing them to swim faster. We know that dolphins and sharks are not closely related, and they did not inherit similar body shapes from a common ancestor. Their streamlined bodies, dorsal fins, and flippers are the result of, notice this, convergent evolution. So they didn't inherit these from past common ancestry. They're not homologous, is what the evolutionists would say. They say that these creatures, because of their environment, evolved the same features independently. Wait a minute, evolution's blind. Why are they evolving the same features independently? I mean, there should be just unlimited features to evolve through blind chance, random process, even though we now know based on recent literature that mutations aren't so random. I could go on all day, so I'm going to have to choose to yield there. Go ahead.
I'm not sure if Luca has a...
sorry, can you repeat the question, please? Because
it was probably specifically for Donny. It's why do you think speciation occurs in other created kinds but not humans? So more.
Okay, that's nothing I can say there because I came from a different point of view. I think that speciation happens in other kinds as for humans. I do not think that a kind it's an actual division. So yeah, let's.
If I could say one thing, Luca, my definition of kind is groups of organisms, independent groups of organisms that trace back to a common genetic pool. Right?
Yes. Okay.
So what that means is variations within a kind, they may or may not be able to reproduce due to how much they've changed. Let's say some are not compatible anymore. I would predict that in vitro they're probably compatible. But the point is, we have been able to do that with humans. The human baramin, through genetics, we can trace them all back to one single genetic pool, which means that's a created kind. And then we would have different genetic pools independent from other genetic pools that we would say is a created kind. So determining what is a created kind has to do with us determining what is and what isn't related based on groups going back to a common ancestor, and that common ancestor not being related to any other groups. Does that make sense?
Yes, but for what I can understand, we can make even a hybrid from humans and pigs, of course, an hybrid with very little of our genome for obvious reasons, but you can do it. So I cannot see that.
A pig and a pig and a human.
I've seen that with my 2 eyes. You can get a human heart inside a pig with that process. And
but you can't even
did that process myself, not on a pig, of course, on a nematode, but I did it.
But you can't even get human sperm and a chimpanzee egg or vice versa. You can't even.
You can not with natural means, but it's quite natural. Once you get to speciation, it's very hard to get a viable offspring, but I think you can get to an hybrid. We are less different than other species that can, but I just think that no one does it for moral reasons. I will think that something like that will be a monster. I cannot even think about it.
Well,
Just think other from a human. It
yeah,
will
I
be
would say that's evil. But if they're forcing this and manipulating this in the lab, then for one, as you said, there's not going to be viable offspring, and it's just going to be some type of monstrosity that's formed. I don't, you know,
well. Yes, you need just to get over. I think the protection of the egg, for what I can understand. So yes, you can get to an hybrid. It's viable, but no one's ever tried it. They tried it in Russia, for what I can tell, but it was with very outdated technology, like at the beginning of the 12th centuries. So it was not possible at that time. So better that way, if you ask me.
well, I just wanted, and last thing I'll say, Matt, you can move on because I think we're going to be going to the after show soon. I just wanted to give a working definition, because a lot of evolutionists will kind of scoff and criticize, like they don't know what a definition of a kind is. And I think that works. That's the one we've used here at Standing for Truth Ministries, is groups of organisms that can be traced back to a common genetic pool. Because that original genetic pool, which I guess you could call the baronome, would be that original genetic pool that was created and front loaded with pre-existing heterozygosity from the created heterozygosity standpoint. So if we can take groups of organisms, determine what is and what isn't related, and trace them back to that original genetic pool, that's us identifying genetic, well, for one, boundaries, but created kind. So I just wanted to clarify that, Luca.
Okay. We are a little past time, but I'll give one more question, and then we can move directly over to the after show. I'll save all the questions that didn't get asked, and that way, if Luca or Donny end up going to the after show, they can answer them there. So the question is for you, Luca. What is the evolutionary explanation for the jump from asexual to sexual organisms, since sexual organisms require multiple organs and systems to work harmoniously?
Okay, you can look it in plants. I just love plants. As I said, I was cloning plants when I was like 18, 19 years old. Plants, modern plants, can reproduce in a lot of ways. But let's take a plant that can reproduce sexually. But you can get that plant to reproduce in other means. So it was a thing like that. You will have in the evolutionary history something that can reproduce in both ways. Like it can reproduce without the sexual part, but also with the sexual one. I remember a good explanation on that, on why there was the difference between egg and sperm, because you can find two kinds of strategy to survive. Be very small and agile, so produce a lot of those kind of cells like sperm, and to be big, have a lot of nutrients inside the cell, and wait for another cell to find you. So natural selection will naturally go in that direction to get to those two extremes, and this is the beginning of sexual reproduction, because it's a very important thing, because without sexual reproduction you can just have some mutation, but the most part it's just identical twins over and over and over. And sexual reproduction is much better if you want to evolve.
Sorry, I was on mute. All right. Any comment that you want to make?
You know what? I'll refrain because I've talked a lot for this Q&A. Luca has been a great guest, a fantastic interlocutor. I appreciate all the prep you did, Luca, to make for a great debate. So I'm going to give him the last word, and we'll make that the final question then. So
great.
Yeah.
Go ahead, Luca.
Nothing to add. It was not very clear, but it's a very difficult topic. It's not like you can get a complete answer in some minutes. So maybe next time.
It's all good. All right, everybody. John, post the link and everybody can head over to the after show. And thank you all for coming here, joining. This was great. Make sure to hit that like button if you haven't already done it. And
I will.
all right. Good.
I did.
Smash it.
give it a good punch.
Yeah. And we will see you guys soon. Donny, if you want to help close it down, you've done
All
this a
right.
lot more than me.
Yes. Great job moderating, Matt. You've moderated my last two debates because I can't really moderate my own debates, right? Unless I clone myself somehow. Maybe through evolutionary processes, I'll be able to do that. So, Matt, you've been doing a fantastic job. And Luca, I appreciate you doing this debate. This was awesome. And it looks like there's some good feedback from the chat. I relatively don't look at the chat while I'm debating because I find it's a bit of a distraction. But when I rewatch it afterwards, I always get a kick out of reading the comments. And I hope everybody enjoyed, because here on Standing for Truth, you know, we pride ourselves in the fact that critical thinking is important. And that's why we host these debates. I believe we've now hosted 160 or 161 debates. And, you know, we like to get together with people of different opinions and views and discuss those things. So I appreciate, Luca, you doing this. Luca, we're going to be having you on a couple more times, I think, in the next couple months for debates. So, yeah, to the audience, thank you so much for tuning in. I'm going to let Matt and Luca get out of here. And there's going to be an after show. And as I usually have been doing, I'm going to come back just for a minute, just myself, and just go over some reminders, guys. So God bless. Thanks again, Matt and Luca. And
see you soon, guys. It was a pleasure, really.
all right, there we go. Another one in the books. As a ministry, as I pointed out, about 161 debates now on all different types of topics. My personal debate number 90. Now, a bunch of people are like, oh, we thought you had way more debates than that. 96. Well, you know what? If we were to count all of the impromptu after show debates, then yes, probably a few hundred. But about 96 formal debates. Luca was a ton of fun to debate. We set this a couple months, and he had some time to prep, bring some good arguments, which resulted in some good discussion. So, guys, we are working pretty much all throughout the day on our upcoming human evolution book. Okay, this is an important topic. Matt and I, we got the thumbnail ready. We wanted to do it sometime after this debate on human evolution. So probably in the next week, we are going to be dismantling the arguments from Gutsick Gibbon that she made on human evolution in a video with, I believe, Seth Andrews. So that's going to be fun. Matt and I, our commentary is probably going to be 3 or 4 hours. So that's going to be a long one, but we got a ton of data. Then I'm also going to be giving a handful of presentations on human evolution. because during a debate, obviously, you can't go through all the slides and data. So I'm going to have really comprehensive presentations for everybody. So again, reminders, guys, the fun continues. We've got event after event after event. The fun continues tomorrow, this time a debate on the nature of God. Is oneness biblical? Kelly Powers and Rodney Smith, both of these gentlemen have debated this topic before. They know what they're talking about. They're well studied. It's going to be fun. It's going to be fun. So make sure you are here for that. Last night, what a fantastic debate. Actually, not last. The days just fly by. They blend together. 2 nights ago, Call Me Emo and Dr. Kenthoven. Check the website too, standfortruthministries.com. We've got a section now, the Evolution Debate Challenge. And I'm doing my best to fill that up with all of the, I guess you could say, episodes in that series. Been a ton of fun. I knew it'd be fun. I just didn't know it'd be this fun. I'm glad we've had so many people requesting debates. Then next week, this is going to be the big one, John. So get ready for a wild after show. Is evolution a reasonable scientific theory? Tom Jump and Dr. Kenthoven, both of them have had what seems to be hundreds of debates. So that one is going to be coming up. And that one is going to be a ton of fun. Also, I'm excited for this. We're going to have a solid presentation lecture. Dr. Ken Colson is going to be back. Dinosaur soft tissue. So critics, bring the best objections you got, because you got questions. We've got answers. So, that being said, just having a look at the chat here. Truth Defenders, good to see you, brother. God bless you. Says, hit that like button, folks. Shalom. Yes. Smash that like button. Truth Defenders, I appreciate that. I appreciate the feedback. Guys, leave a comment. Tell us how you feel the debate went. What you agreed with, what you didn't agree with. It's an important topic, and any willing evolutionists want to take the challenge. You can debate myself. You can debate really anybody from the team that would be interested in debates, of course. So that being said, let's head over to Logical, Plausible, Possibles channel, Probables channel, and enjoy an after show. John could probably guess it. I'm going to get some food and a coffee, then I'll be there. So it should be fun. And until we meet again, everybody, God bless. Thanks for tuning in. Stand for Truth is out.